Thank you for joining us for our next video called Protein Synthesis. We're going to start with an overview of the steps in protein synthesis, which you should already be familiar with, but we're just going to make sure everyone's on the same page. So first we have transcription, which is the production of mRNA, rRNA, or any of the RNAs from DNA template. And then we have RNA processing, which only occurs in eukaryotes, which is where we're going to make some modifications to the RNA so that it can then be utilized by the translation machinery. And then we have translation, which is where we take the RNA code and turn that into a protein. So we're going to start off by talking about the differences between transcription and translation in prokaryotes versus eukaryotes. So you'll notice by looking at the picture here that in prokaryotes we just do transcription where we produce RNA, and in this case we can show the mRNA, and then we'll do translation, which is where the RNA is read by the ribosome and produces a polypeptide, and that polypeptide will then be folded and become a mature protein. And in eukaryotes, because the eukaryote has a nucleus, you can see that the DNA is inside of the nucleus, as well as the RNA that's produced starts off in the nucleus. And at that point we call this a pre-mRNA because it still has some processing to undergo. And then we'll have RNA processing occurring where we make some changes to the RNA, and I'll describe the processes that are involved there a little bit later. And then we'll have a mature mRNA molecule, and that mature mRNA can then exit the nucleus and it will find its way to a ribosome where it will be also read just in the same manner, similar manner to the way that it's done in prokaryotes. And then we end up producing a polypeptide, and then we're finished with protein synthesis. So keep watching for the next video because we will talk about the details of transcription. There are 3 steps in transcription. They are initiation, elongation, and termination. And some vocabulary that you will need to know by the end of this video are the promoter, transcription factor, transcription initiation complex, and also the TATA box. We will talk in detail about what each of those things are and how they work together in transcription. Remember, the goal of transcription is for us to produce a mature mRNA molecule. And we're going to be using that mRNA in another process later called translation. So first, the DNA sequence where RNA polymerase will attach is called the promoter. So remembering that you've learned before that RNA polymerase is a molecule that will read the DNA template and produce RNA. That's why it's called a polymerase, because it makes a polymer. So we're going to attach in a region called the promoter region. That region is found at the very beginning of a gene, and it's right here on your notes. This is our promoter. And it's underneath that bubble, which is our RNA polymerase. So we'll just go ahead and mark that. All right, and then at the end of this sequence, we have what's called a terminator. And the terminator, kind of obvious, is where we stop transcription. And so the terminator will be right here at the end of the gene. So during initiation of transcription, RNA polymerase is going to join with the DNA template, and then it will begin to unzip the double helix by breaking those hydrogen bonds. So that's what's happening inside of this little bubble, is that the double helix is being unzipped by hydrogen bonds breaking. So that's what RNA polymerase is doing right here, and that's initiation. And then elongation is the process of the mRNA, in this case, being elongated or made longer. And that continues until you reach the terminator sequence. And once the RNA polymerase reaches the terminator sequence, it will dissociate or fall off, and then we have a completed piece of mRNA. Of course, we've left out some details, but this is the basic overview of what's happening. Really, one thing we need to make sure we understand is that when we're producing an mRNA, we're actually doing this according to base pairing rules. So that's the overview. Let's just keep going. In your notes, what I've done here is just taken some of the pieces of diagrams from your text so that you can annotate them just to make sure that everything makes sense to you when you're reading your book. So let's just talk about what we see here. So first of all, we have the TATA box. It's called the TATA box because it actually has the sequence TA, TA, AA. And then of course this is my template strand, which is being read by the machinery, and then we have the other strand. All right, this is our promoter region. And remember, the promoter region is the region where RNA polymerase can bind. And then the start point of the actual transcription is somewhere downstream of the TATA box. And by downstream, I mean later in the sequence. Your template strand of DNA is the one that will produce the mRNA, and it's actually being read 3' to 5' because remember, we always produce new nucleotides in the 5' to 3' direction, and they have to be anti-parallel with each other. All right, what are these purple bubbles here? These are transcription factors. You might remember talking about cell signaling and learning that one of the possible responses of cellular signaling is that molecules become transcription factors and encourage genes to be expressed. That's what these things are. These are molecules that were produced with an effort to encourage gene expression. All right, so the transcription factors will bind to the DNA, which then allows the binding of RNA polymerase. You can think of this as sort of a landing pad. Helicopters land on a specific spot that's been marked a certain way. So that's what's going on here is the transcription factors are binding and encouraging the RNA polymerase to bind at that very spot. So then again, our RNA molecule is going to be produced in the 5' to 3' direction along the template strand, which is read in the 3 to 5 direction. All right. In the next segment, we'll talk about more detail of how exactly RNA polymerase is able to copy the DNA template. Let's talk briefly about how RNA polymerase is able to produce RNA. In this diagram, we're looking at RNA polymerase up close. So this really large blob is our RNA polymerase. And of course, you can see the DNA template and the RNA being produced. We can see that we're reading our template strand, which is this dark blue, from the 3 to 5 direction. And here's my non-template strand. And of course, we can put whatever bases here we want. I think there's a naming convention where they decided that C's and G's would be curvy and A's and T's would be pointy. I'm not sure if there was a committee or what happened there. But you can see that they match up with each other. So if this is an A, then this would have to be a T down here. And then that's how the original strand was stuck to each other. So A with T, T with A. And remember that in DNA, we have thymine, but in RNA, we would use uracil. So in a moment, when I start writing U's, that's Y. So there's my DNA template. The hydrogen bonds between the base pairs are temporarily broken by RNA polymerase. And then RNA polymerase is going to place these RNA nucleotides in order based on these base pairing rules. A with T, U with A. Remember, we don't use T. No thymine. Right, like this. So this is going to be a U. And then you see this U is coming in here, and it's going to bind with that A. Now this all is happening because of binding affinity, meaning they like to bind with each other. It has to do with chemistry, forming of bonds, a little bit of physics, and how exactly does that happen? Well, diffusion. Things move around and they end up in the right place. They stick together a little bit like a magnet. This is the direction of transcription overall. So you can imagine if this whole entire molecule were moving in this direction, it would be traveling along the template strand from 3 to 5, producing our new molecule in the 5 to 3 direction, adding new nucleotides to the 3' end of the molecule. And that's elongation. And then the next step is termination, which is meaning stop. How does it know how to stop? Things are a little bit different between prokaryotes and eukaryotes. So specifically, in bacteria, the polymerase will stop when it reaches the terminator, and then mRNA is ready to go. It's finished. It doesn't need any further modification. So I'm just going to write that down. It's ready to be translated. In eukaryotes, however, we have to make some changes. So we have to be prepared for those changes. So RNA polymerase II, which is, another polymerase, is going to add the poly-A tail. It's actually called the polyadenylation signal sequence. And what that means in plain English is A. It's going to add several 10 to 35 nucleotides on to the end of just A, adenine. This is telling the molecule that it's ready to be processed.